Shall we get started, Jean?
Yeah, so Ben, do you want to introduce yourself just after me, or right?
Yeah, sure.
Okay, so let's start now. Hi, good morning. I'm Zhang. I'm a postdoc at Center for the Physics of Biological Function. I work with Professor Weiner mainly on intracellular phase separation. So Ben?
And my name is Ben Weiner. I'm a grad student in the physics department, just finishing my 4th year, also working with Professor Weiner, and I grew up in Cincinnati, Ohio. And I will let Jean take it away for the first half here.
Okay, thanks, Ben. So today, Ben and I will be leading the discussion session for Professor Weiner's lecture on intracellular phase separation and magic numbers. So Ben and I have went through many of your questions, and we found that most of the questions can be separated into 4 categories. Some of the questions are purely about biology. What are the biomolecular condensates and what are their functions? Some of questions are purely about physics, asking about the minimal models for biomolecular phase separation and the Flory-Huggins model. And then there is a set of questions asking about the connection between biology and physics, asking about at the molecular level, what are the interactions and how these molecular interactions give rise to phase separation of biomolecules. And finally, there are a set of specific questions asking about the magic number effect, and also asking about different methods of modeling the intracellular phase separation. So I will be covering the first 2 points today, and later on, Ben will be covering the last 2 points. Okay, so before I start with the biology part, let me first begin with this, sharing some of my experience with you with regard to this question. So this is a question raised by one of the summer school students, how to make the most out of scientific papers when a lot of the content in it is hard to understand. So this is a common situation, especially for people who just started their research at the initial stage of research. You'll find that many papers are pretty hard to understand. And I personally also have the experience when I was trained as a physicist, and when I first started to do research in the biophysics area, I found most of the biology papers very hard to understand. So here is what I will do. I will try to search for terminologies in Google or in Wikipedia. If I found these terminologies in the paper I don't understand, I'll just grab some basic ideas about these terminologies. I'll also try to find experts who can give me an introduction about the topic. Or nowadays, we can actually search online. Maybe there is a talk by one of the authors in the paper, or also maybe there is a talk that has a very related topic to that paper. So that's what we can do. So after that, if I still find this paper is just hard to digest, then I'll just keep reading this paper and then keep reading more papers and try to revisit this paper later on. And most of the time, I find the paper becomes easier to digest, say after a month or so, when my understanding builds up with time. So these are the experience I had, and I hope maybe it's helpful to you as well. So with that, let's start with this biology part. Let's try to learn what are the biomolecular condensates together. So a recent major development in cell biology in the recent decade is the discovery that eukaryotic cells are host to a multiplicity of non-membrane bound organelles. And these non-membrane bound organelles are what we are talking about. These are the biomolecular condensates. What are the biomolecular condensates? These are just condensates of condensates of molecules specific molecules coming together with no lipid membranes surrounding them. So these are biomolecular condensates, literally. And so in this figure I'm showing here, there are lots of different types of biomolecular condensates, and I'm not going to go through all of them, but we can talk about some of them. And in the first figure, this is the elegant embryo. This is anterior and posterior, and these green puncta here, these are the P granules. P granules are made of RNA molecules and proteins, specific type of proteins, and they mostly stay in the posterior of the embryo, and later on they will end up in the germline cells, and that's why they are also called germ granules. Now, the biological function of these P granules is still in a way unknown. There are speculations saying that maybe these granules, although in the cytoplasm, may adhere to the outer membrane of the nucleus and be gating some entrance of export of the RNA molecules. But these are all speculation. And so you see that this is a very new field, and there are certain things that are still unknown. That's why it makes this field exciting and also challenging. And okay, so in the second part of this piece, this figure, this is a signaling cluster. So a signaling cluster means that it forms in response to signal from outside of the cell membrane. And this cluster is made of a bunch of different type of proteins. And they form underneath thing. It is a stress granule. Stress granule by name, you could see that it forms under stress, and so and it is made of mRNA molecules and also proteins. It forms stress. The stress granule forms when there is a stress. It protects these mRNA molecules inside, protecting them from degradation. So when the stress is gone, the mRNA molecules in the stress granule get released into the cytoplasm, and then the cell can immediately translate these mRNA molecules and making new proteins to repair from the damage from the stress. And so this one, it's a nucleolus, and nucleolus is a biomolecular condensate that is inside cell nucleus. It is made of RNA molecules, proteins, and also involves some part of the DNAs. And it is majorly, its biological function is majorly for ribosomal biogenesis, which means it assembles ribosome subunit, and ribosomes are used to translate mRNA molecule into proteins. So, and this figure shows that this ribosome, this nucleolus is liquid-like. It can merge with each other and form a new spherical rounded liquid droplet. Okay, so after talking about all these biomolecular condensates and their functions, now let's zoom in a little bit. These biomolecular condensates are mostly of micrometer or sub-micrometer in size, and if we zoom in, there are molecules inside which are interacting with each other. So for example, this picture is resembling these signaling clusters, which has a bunch of different type of proteins inside. These proteins are multivalent. Multivalent means it has multiple repetitive domains that can bind to each other. Say this yellow domain binds to this green domain between these two proteins, and you can see there are 4 domains and there are 2 domains. So the binding is multivalent, and the binding is usually weak and transient, but because of the multivalence, it brings all these molecules together to form a condensate. And here is another example, involves intrinsically disordered proteins. Ben will be talking about more about intrinsically disordered proteins. I'm just going to say that, say this molecule has two RNA binding sites that can bind to RNA. So one RNA can bind to multiple this type of proteins, and then all these proteins have this sticky tails of intrinsic disorder regions hanging out and sticking to each other, which then drives the liquid phase separation of and formation of this biomolecular condensates. Okay, so that's the first part, and I will pause for questions. Yes. Okay, so Ben, where can I find the questions? All right. So, okay, so there is a question. I have a question about the dimer degeneracy and wait, this is too fast. Okay, I have a question about the dimer degeneracy and the clustering tendency as alluded to in the paper about magic number effect. Oh, wait, I can't catch that. Ben, hi Ben.
Hey there. Okay.
All right, so I can catch that. I'm sorry.
Yeah, it flipped into the answered category. So we're done. No.
Oh, we are done.
No,
All right, great. Okay, so we don't have actually questions. I can move to the next part.
I think I'll talk more about this question when I cover the magic numbers section.
Yeah, that seems to be a question about magic numbers.
There's also this question about signaling molecules. So just at a very like high level conceptual level,
yeah,
phase separation can concentrate things in specific places, which can certainly accelerate the signal passing through some of these pathways. So one example is that it was recently shown here at Princeton that certain forms of the activation of T cell receptors and immune signaling actually takes place when a phase-separated droplet forms on the membrane of the cell. And you actually need phase separation to concentrate all those signaling molecules for the immune signaling to take place. So that's just one example.
Yes. T cell activation is indeed a signaling cluster, involves the signaling cluster formation, even though I'm not an expert on these biological questions. Okay, so maybe I should move on to the next part. Okay, so now let's switch the gear and talk about physics of phase separation. So let's start with maybe just a simple lattice model and trying to develop some intuitions. So in statistical mechanics, we know that a system, when it's in equilibration, it has its free energy at the minimum. The system always has a tendency to minimize its free energy when it's out of equilibrium. So if we have a system like this, if we have a lattice, each lattice size can be occupied by a solvent molecule or a polymer, then if we try to mix a solvent and a polymer into a solution, what will happen? If there is no energy or there is no interaction between the polymer and solvent, or the interaction between the polymer and solvent is just a constant, doesn't matter if it's between the solvent and the polymer or between the polymer and the polymer or solvent and solvent. Then we know that in order to minimize this free energy, the system wants to maximize its entropy. And entropy means randomness. So in order to maximize entropy means the system wants to be as random as possible, which will then result in a homogeneously mixed solution, a solution of polymers. Then what can we do to make the system phase separate? Well, because we have a competitive term energy here. So if the system have interactions between, say, polymers, between solvent, and between polymer and solvent. And if the system, in the system, the polymer likes polymer, and the solvent likes solvent, but the solvent do not like polymer that much, which is just like, say, if we have water molecules are hydrophilic, they like each other. And then if we have oil molecules, and the oil molecules, they are hydrophobic and they like each other. But the hydrophobic water molecule do not really like the hydrophilic water molecule do not really like the hydrophobic oil molecule. Then that makes an interaction energy in this form. And we know that when this happens, the system will want to phase separate simply because these polymers, they like each other, so they like to stay with each other and forming this dense phase with high polymer concentration. And then the solvents also want to stay with each other, so they form a dilute phase with low polymer concentration. So these are the intuition. So having developed the intuition, now let's make it more quantitative and move to Flory-Huggins theory. So for simplicity, we'll deal with the polymer of length 1. So it's really a particle here. So for this system, what do we want to do? We want to find the free energy, and the free energy of mixing, how much free energy is costed to mix the two solutions into a homogeneous system. And to find the free energy, we want two terms of the entropy and energy. Now for this system, because there is only one configuration, all the solvents are here and all the polymers, polymer particles are here. So one configuration means entropy equal to the Boltzmann factor times log of the configuration. Configuration is 1 means entropy is 0 here. Now for this system, then the entropy is KB times log of the number of configurations. And here we have many different configurations because we can choose, essentially we can choose NP number of polymers sites outside the total number of N sites. So that is the total number of configurations, and this is the entropy in the system. And then we want to calculate the energy in these two systems, and the energy is really about how many pairs you have with both of the side with polymer and polymer neighboring sites, with polymer and polymer, or with solvent and solvent. So let me take an example here. We have NP polymer here. Each of them have a neighboring site Z. Then each polymer will have NP times, will have Z bonds, but this bond is shared by two neighbors. So each polymer have effectively Z over 2 bond, and NP polymer means we have NP times Z over 2 bonds. So that's why we have this term. And similarly, N minus NP is the number of solvent. So then the number of solvent times Z over 2 is the number of total neighboring pairs for the solvent molecules. And similarly here in total, we have number of N side times Z over 2 number of neighboring pairs. Now for each pair, we have this amount of probability to have both of the side to be polymer and polymer. And then this amount of probability to have both of the side to be solvent and solvent. And then this amount of probability to have the side to be, one side to be the solvent and one side to be the polymer. And that's why we have the binding energy in this form. So having all of these, if we plug into the free energy expression, and we'll find that free energy of mixing per lattice site is expressed in this formula, where phi is a fraction of polymers in the lattice, and then chi is the interaction parameter, which involves all the binding energies, neighboring interaction energies, and the neighboring side, also the temperature. So these two terms are coming from the entropy contribution, and this last term is from the energy contribution. And so if you are familiar with the free energy of ideal gas, you know that the ideal gas free energy is C log C, where C is the concentration of the ideal gas. So here it's really like very similar. Phi is the concentration of the polymer, so you have phi log phi. But 1 minus phi is the concentration of the solvent. So you have 1 minus phi log 1 minus phi, denoting the entropy of the solvent. So entropy of the polymer, entropy of the solvent, plus their interaction. Okay, so what if we have the polymer which has a length that is not 1? So it turns out that if the polymer of length is not 1, all the contribution to all the changes we have to make is really just to insert the L, the length of the polymer, into the denominator of this expression. And for details of the derivation, you can refer to this lecture note, which is very clearly written and very well explained. And so essentially, so now we have this. So now we have the free energy of mixing here for the polymer solution, and how can we make use of this to find to understand the phase separation of the polymer or phase separation of biomolecules. So we know that there is an interaction parameter chi here, right? And then we know that if the interaction parameter is 0, then it's really entropy contribution. And if we plot this expression, we find that the curve is like this, which is concave. And if we have, when we increase the interaction, which means that if we increase the polymer-polymer interaction, we make the polymer like polymer and makes the solvent like solvent and makes the polymer and solvent do not like each other, then we will increase the chi. And when the interaction parameter chi is above certain threshold, then the free energy will have a regime which becomes convex. And so what is this convex part mean? If we have a convex part in the free energy, then we know that if we have a system, if we start with a system with the global concentration of the polymer to be phi 0, then the free energy, if the system is homogeneously, if it's homogeneously well mixed, it will have a free energy of this one, because that is how we derived it. However, the system can also try to phase separate. It can phase separate into a dilute phase and a dense phase with much higher concentration of polymers. And if the system phase separated into these two subsystems, then it turns out that the free energy per lattice side for the combination of these systems, the subsystem, is here at the black dot. So you could see that the free energy of this phase separated system is lower than the free energy of the well mixed state. And that's why the system prefers to phase separate, because we always want to lower the free energy. And so that's how we can draw the phase diagram. So if we have the temperature or inverse of critical, inverse of the interaction energy that is high, then we know that the system always want to be in the well mixed state because the free energy is concave. And when the temperature or the interaction parameter chi is below certain critical point, then the free energy becomes concave, and then by phase separating into a dilute and dense phase, the system will be able to lower its free energy, and then we can just add 2 points, the dilute phase concentration here and dense phase concentration here, and making a phase diagram for the system. Okay, so that's the part about the Flory-Huggins model. And I'll pause for questions. So Ben, coming up.
Hi, still here.
Hey. Yeah, so I don't know if there are any questions you have already answered.
I've been typing up a bunch of answers to simple questions.
Yes.
Let's see here.
Why do physical systems want to have the lowest free energy possible in the first place? Okay.
It's a deep question.
Yeah. Well, that's thermodynamic laws, right? There are 3 laws in statistical mechanics.
I think that for the most part in the day-to-day people, this is just sort of an axiom that seems to be true about the world. And it's a principle that you can study the consequences of, but understanding where those principles come from is I think, harder. That's what I would say. I don't know. Do you have any comments about the hydrophilic or hydrophobic gene?
What makes something hydrophilic? Well, I guess hydrophilic means you have polar, like you have electron density that is. So if you have carbon and carbon, then if they share electron, the electron will be kind of shared equally by both of these 2 atoms. So you have kind of hydrophobic type. But if you have hydrogen and the oxygen, then the electron density will be shifted to one of the atoms. And then it becomes like polar or dipole-like, then it becomes hydrophilic, right? How do phase diagram plot? How is the phase diagram by Flory-Huggins equation? So if Flory-Huggins theory, so I'll say in practice, in experiment, what you need to do is always just say, try to tune the temperature, and then try to measure the dilute concentration and the dense phase concentration. So then is really just find the dilute concentration and plot a dot here at the temperature and concentration, and then find the dense phase concentration and plot it here, temperature and concentration. So then when you change the temperature, you will automatically have different points on this curve, and eventually you'll be able to find the whole line here. That's how in practice the phase diagram is plotted. If you have if you have a theory, then you apply the convex Hall analysis. Something like here, you always find the lowest line that are tangential to your free energy. And so this free energy will change, the shape of free energy will change with respect to temperature or with respect to this interaction parameter chi, right? So then for each given temperature or interaction parameter chi, you do the convex Hall analysis. So convex Hall analysis is really right now very common, and you can find codes in MATLAB or something else, just to find the lowest points in this curve that are tangential to this curve. And then you find the dilute phase concentration here and dense phase concentration here, and these 2 points enters here with this y-axis to be the temperature. So then you can change the chi or the temperature here, and you will have another free energy shape, and you do another set of convex Hall analysis, and you may find another 2 points on the line. That's a way to find the free energy. Find the phase diagram. What are the practical use of phase separation? Practical use of phase separation out of the cell in daily life. Phase separation. Well, I guess there are many. Ben, you have any comments on this? Phase separation should be.
Yeah, if you go hang out with your chemical engineering colleagues across campus, there are a lot of people who've been doing polymer physics sort of for industrial uses for many decades now. And you can mix together like blends of different types of polymers with different interactions, and they will phase separate into all sorts of interesting structures that you can use for manufacturing plastics or for designing materials with certain properties and things like that. So yeah, there's definitely like a huge set of industrial applications for phase separation, especially with of polymers in particular. Do you think that? Oh, go ahead.
Yeah, maybe one of the last question. Which one do you pretty sure am I? Oh, too many questions. Am I thinking that phi1 is the concentration outside? Too many questions. Oh, convex hull. Okay, someone is HULL. Do you have any suggestions for books that help connect calculus and mathematics to physics? Too many questions. What can we do? Ben, I think we should move on, but yeah, we can come back to the questions later. I'm sorry. Okay. You should move on. Yeah, otherwise we won't be able to finish.
Great. We'll move on. Some of these things will become more clear as I talk, hopefully.
Yes, yes. Okay, so let me stop sharing my screen.
Great. Hi, everyone. So thanks for being with us this morning. And so, as June said, I'm going to be addressing questions that fall under sort of two thematic categories. The first is really connecting biology and physics, and then the second is talking in more detail about the magic number effect, and some of your questions about more general computational methods for studying phase separation. So June's introduction to Flory-Huggins is actually a great jumping off point, because the Flory-Huggins theory is really good science, but it's also, at this point, very old science. These papers came out almost 80 years ago now. And so actually the reason that we need to go beyond Flory-Huggins theory today is exactly because the biology is teaching us new physics to combine with the polymer physics that June was talking about. And so in particular, there are sort of three big things that make intracellular phase separation much richer and more complicated than the sort of homopolymer phase separation that Flory and Huggins were thinking about in the 40s. So the first is just the components are much more complicated. So instead of like a very simple homopolymer where every single component is the same and interacting with each other in the same way, we have very complicated proteins, often dozens or hundreds of types of proteins interacting with each other and with other small molecules in complicated ways. As people have alluded to in the chat, cells are always out of equilibrium, and you have to worry about this in various ways. And in the cell, you're not thinking about an infinite homogeneous system, right? There's lots of variation in both space and time for any particular cellular component that you're interested in. So I will sort of address these last two things first. So we had some questions that were just asking us to explain in a little bit more detail what we mean when we say that cells have spatiotemporal control of biological processes. All this means is that the concentrations of various stuff in the cell are going to vary in time and space, and the cell wants to control how that happens to survive. So one cool example is that there's increasing evidence now that phase separation might be used for gene regulation. So basically, there's an idea that cells are actually condensing droplets at specific parts of DNA to concentrate transcriptional machinery to transcribe that DNA into RNA and eventually make protein. And so that's a process that has to happen at the right time and on the right place in the DNA. And that's what we mean by spatiotemporal control. Now, people have pointed out very rightly, and you've probably had this drilled into your heads in the summer school, that life is out of equilibrium. If you're equilibrium, you're dead, right? And of course, cells are constantly growing and dividing and reacting to their environments. This is immediately obvious if we look at these images of HeLa cells, where the little yellow dots are P-bodies, which are membraneless organelle. If this were a simple homogeneous equilibrium system, there would only be one large dot, right, in that sort of very simple case. And just the fact that we can see lots of these dots together tells us that something more complicated is happening. But you can really think of equilibrium as a matter of time scales and of spatial scales. So it might be that the cell is dividing on a time scale of hours, and it might be that the concentrations of things in the cell are varying over microns. But if one of these little droplets can form in seconds, and if it's only, say, tens of nanometers or hundreds of nanometers instead of microns, then you can actually probably learn a lot by just studying the equilibrium physics of just one of these droplets. That's an example. And so this is why, you know, there are a lot of cases where even though cells are out of equilibrium, we can, as long as there's like a good separation of time scales, you can often learn things by using equilibrium physics on a smaller part of the problem. So that's sort of the side, which is that phase separation is happening in cells which are out of non-equilibrium. There are also a lot of non-equilibrium processes which affect phase separation directly. And this is a really rich area that people are still exploring. It's discussed a lot in the review paper by Brangwyn et al. So, you know, one thing that you can imagine a cell doing is if you have a droplet, you can modify the components that are in that droplet through some post-translational modification, for instance. That's certainly a non-equilibrium process. There are now a lot of cool ideas about how cells can actively burn ATP to fluidize the components of these droplets and maybe prevent the formation of pathological gels or solids that could actually be, you know, very dangerous for the organism. And as I said, I'd say that this is still a really active area of research. Next, I'll talk just a little bit about IDPs, and June actually already covered this super well, so I'll be quick. I think people are just getting a little caught up in some of the terminology here. There's a lot of jargon. An IDP is just an intrinsically disordered protein, and we say it's intrinsically disordered because it doesn't fold into a unique 3D state. And so, you know, instead of having some like nice folded protein that you might be used to seeing when you hear about the protein folding problem, you just have something that actually looks a lot more like a polymer. And it's free to wiggle around and explore lots of shapes. And so we call the 3D shape of the protein its conformation. And because it has many shapes, we say it has conformational heterogeneity. And so that's just terminology. Now, people also had lots of questions about interaction domains. And I'd say that this is really a huge part where the richness of biology challenges us to go beyond the traditional Flory-Huggins model. So, you know, in the interaction domain, to put it super simply, is just a unit of the polymer that is sticking to the other units. So if you have like a classical sort of Flory-Huggins homopolymer, you just have a bunch of monomers which all just stick to each other in the same way. But proteins actually are made of a bunch of different amino acids, which are all interacting with each other in complicated ways. So you have this whole hierarchy of interactions, from relatively long range electrostatic interactions between charged residues, all the way down to very short range pi-pi stacking, which can only happen between specific aromatic amino acids. And you know, this is just these are just interaction domains on the level of single amino acids in an IDP. You can also have longer chains of or longer parts of proteins, which are themselves interacting motifs, or you can even have much more complicated, you know, sort of peptide bonds where each interaction domain is itself, you know, is a pretty complicated protein that's linking together. And so systematically dealing with all of these interactions and deciding which are important for what aspect of the behavior when remains, you know, one of the major challenges in the field, and a really active area of research as well. We had questions about performing experiments and connecting what's happening in vitro or in a test tube to what's happening in vivo or inside cells. And I have to say, you know, I'm a theorist, so I don't want to get in too much trouble by saying too much about experiments. But what I will say is that people work really, really hard to match the conditions in vitro and in vivo. So of course, you're going to try to do your experiments at the same temperature that the cell would be in. You want to try to match the concentrations as much as possible. Right? You might be able to see that some protein phase separates in a test tube, but if it only does so at concentrations that are sort of 10x the physiological concentration, that may or may not still be interesting, but it's probably less relevant for biological function. And as well, people pointed out in the questions that there's a lot of, you know, the interiors of cells are very crowded. And so indeed, in experiments, people will actually add molecular crowders to try to replicate this in the test tube. Another way to try to understand what's happening in vivo is to actually put in, you can actually put synthetic phase separating systems into cells and then control them with light. So this is a technology called optogenetics. And the idea is that you can genetically engineer cells to express these proteins that only phase separate when you turn on the light. And so this is actually in vivo, but because it's something that you put into the cell and you can control, it has a lot of the nice, replicability and controllability that you would get from an in vitro experiment. All that being said, experiments are very challenging, and the real world is very complicated, and things can still go wrong. I saw a talk last year where people were very confused because they had a protein that they thought phase separated, and then all of a sudden they came into lab one day and there were no droplets forming anywhere. And after a lot of work, they discovered that what had happened is that they had switched the brand of glass slide that they were using to do their experiments. And it turns out that the old brand of glass slide had like some sort of template that was allowing these molecules to stick together and phase separate, which is presumably not something that's happening in the cell. So even under the best of circumstances, it can sometimes be very difficult to connect what's happening in vivo and in vitro, but it's something that people think very deeply about when they're doing experiments. And then we got some questions about the role that temperature plays. So the simple physics answer is just that when you change the temperature, you move around in the phase diagram. So you can imagine that if you're in the two-phase region and you have a droplet, and then you raise the temperature, the droplet dissolves. You can also have situations where high temperatures favor phase separation, although this is more common, I think, in industrial or non-biological polymers. So that's the simple physics answer. I think that the role that this plays in biological function is interesting and not necessarily studied very much. One of my favorite groups, one of my favorite research groups, is this group out at the University of Wyoming, where basically they torture water bears for a living, or tardigrades. They're these kind of scary looking alien guys. And these tardigrades are incredibly resilient creatures. They can survive extreme heat, extreme cold, being exposed to the vacuum of space, or being dried out and then rehydrated weeks later. And so what they found, is one of my favorite figures in a science paper, is that you can basically, one of the ways that these water bears survive drying out is that the IDPs inside of them undergo a transition to a gel, and this sort of prevents damage to a lot of the intracellular components, somewhat analogously to the stress granules, I think, that June described earlier. And this is actually a temperature dependent effect. Once you go past the glass transition temperature, this stops working as well. And you can imagine that water bears are also perhaps using similar mechanisms as a response to heat shock, right? So you actually, it's very plausible that sort of protein phase transitions are an important response to temperature changes. I'll also say that people do most of their experiments in things like human cells for obvious reasons, but there are lots of creatures that live in more extreme environments throughout the world. So there are these sulfide worms that live by deep sea vents and spend most of their time at 120 or 130 degrees Fahrenheit. And so we know that many extremophiles have different proteomes that can function in high heat. And I don't think people have thought very much about, if these proteins are phase separating and how you design proteins that can phase separate at these sorts of temperatures. It's a very open question. So I encourage one of you to go out and start a lab that studies phase separation in sulfide worms, and let me know what you find. I think that is probably a good point to stop for questions before talking about magic numbers.
Yes.
There's this question about non-equilibrium tuning. So yeah, so like I said, there's an idea that cells are utilizing energy sources to prevent the formation of these gels. Well, I mean, one very simple thing that you can imagine, and I wouldn't claim that this is necessarily the most stereotypical or generic thing, but you can definitely imagine cells burning ATP just to drive transport. And as you drive transport or promote diffusion, you're going to fluidize these droplets to some extent, and hopefully, you know, prevent these things from aggregating in a way that can perhaps hurt the cell, like you might find with a lot of neurodegenerative diseases like Alzheimer's. What else? There's a question about pressure. Yes, pressure is definitely another control variable that you can use instead of temperature. Yeah, you can you know, you can do temperature, you can do pressure. You could some systems have a chemical potential where you're exchanging particles with like a reservoir. So there are lots of different ways to draw these phase diagrams. We have a question about the lower critical solution temperatures. Or no, single phase at the top and two phase at the bottom. Now that's actually the simpler case. Yeah, sorry, let me go back. So that's this question, a single phase at the top and two phases at the bottom. The reason you have two phases at the bottom here is just that this is driven by things like really wanting to stick together. And the colder it is, the more favorable that it is to stick together and minimize that energy. That's like one very simple answer. Although, of course, it's always relative to the entropy, which becomes more important at high temperature and usually promotes mixing. And I think I will go ahead and move on. All right. So the second thing I sort of want to talk about is addressing this issue of specific and non-specific interactions and magic numbers. And so this is reiterating a lot of what Gene said, but basically, you know, a lot of systems that people have thought about in the past with phase separation are really driven by non-specific interactions like oil and water, where, you know, all of the yellow guys and all of the purple guys are interacting all with each other, right? And the same would be true for like the sort of Flory-Huggins homopolymers, where each monomer is interacting with all the other monomers in the same way, perhaps because they're hydrophobic or hydrophilic. Now, once you have proteins or IDPs, you can have much more complicated things where, say that you have two amino acids forming a cation pi bond, right? This can only happen between an amino acid that's a cation and then an aromatic residue. So in that sense, it's specific. You can't just put any two amino acids together and have that interaction happen. So that's a specific interaction. There are also lots of examples of these peptide bonds that we've been mentioning. So there's, I believe that Ned in his lecture is going to make you all experts on the epic Rubisco system, which is important for photosynthesis in algae, so I won't dwell on that. There's another system from human cells called the SimSumo system, where basically, you know, all you really need to know about it is that you have one component that's this blue guy and one component that's this yellow guy, and they come together and they form sort of a one-to-one lock and key type interaction. And so when you have these sorts of very specific and saturating interactions, you can actually have very new physics, which is, I think, counterintuitive from like the old school Flory-Huggins perspective, including the magic numbers effect. And so I will give you guys just a very brief review of what magic numbers are, and then talk about some of the more specific questions. And so the basic setup is that you have two protein types. You have a rigid block with 8 binding sites in this case. That's this blue guy. and then a flexible protein with L binding sites. That's the red guy. And so your intuition from traditional polymer physics with non-specific interactions is just that as you make this, this flexible polymer longer, you should get more phase separation. But in fact, what happens is that there are certain lengths, like L equals 4 or like L equals 8, that are magic numbers in the sense that when polymers have this length, small oligomers, like dimers, become very stable entropically, and this actually suppresses phase separation. And that's what you're seeing here in this picture, where you're going from, you know, in all three of these, you have this rigid block with 8 sites, but as you increase the polymer length from 7 to 8 to 9, you see phase separation, dimers without phase separation, and then phase separation again. And this is actually named after an effect in nuclear physics where certain numbers of nucleons, so protons or neutrons, are much more stable against radioactive decay than other ones. And it's in a very non-monotonic and non-trivial way. And so this sort of magic numbers effect is something that you can really only get from specific interactions, which people, again, did not think that much about in traditional polymer physics, but are all over the place in biology. So we had some questions about the theory and how you model this. And I don't want to get too down in the weeds here with these equations, but basically what I'll say is that what you're trying to capture is that there's a competition between the dense phase and the dilute phase. And so in the dense phase, essentially there's high conformational entropy, which is just the number of ways that this, this red flexible guy can wiggle around. But there's low translational entropy, which is just how much you can move throughout space. So the idea is that you can wiggle around a lot, but you're stuck in the cluster. And so basically, these terms are your high conformational entropy, and these terms have to do with your translational entropy. On the other hand, in the dilute phase, there's high translational entropy because these dimers can float around and go anywhere, but there's low conformational entropy because there are just fewer ways to you know, to form these dimers. Basically, once you're stuck, you know, on one of these blocks, you're sort of stuck there. And so that dimer degeneracy is quantified here, and then the translational entropy is quantified in these terms. And the idea is that if there's phase separation, there are going to be some concentrations where you can lower your overall free energy by putting some of the system in a dense state and some of it in a dilute state. Let's see. We had some questions about the terminology here of sort of like what's the difference between phase separation, gelation, and percolation. So just to give you guys a rundown. So phase separation is what we've been talking about this whole time, where you have a dense droplet which coexists with the dilute phase. And this is a first, if you're familiar with the classification of phase transitions, this is a first order phase transition because there's a discontinuity in a first derivative of the free energy. So that's the density, that's the order parameter here. Now, a gel is just a connected cluster of polymers or proteins. So in our case, the dense phase is a gel, right? Like this is clearly a cluster of proteins. And so gels and phase separation are definitely not mutually exclusive. Now, there are lots of models or lots of physical systems which undergo a gel transition. And it's definitely not immediately obvious like what kind of a transition this is, where you form, you know, an infinite gel, which sort of goes on forever or expands your system. In a lot of the models that we're sort of interested in for this kind of protein physics, it turns out that gelation is actually a continuous transition. It's not thermodynamic at all. But importantly, with a gel transition, you're not going to have these two different concentrations coexisting with each other. If you have a phase-separated droplet and you add more stuff to the system, the droplet gets bigger, but it doesn't get denser. If you have a gel and you add more stuff to the system, the gel gets denser. And we actually show in this paper that the former happens because it's phase separation and not the latter, where it's just a gel transition. And then finally, percolation is something that happens where, you know, if you're just filling up a lattice, at some point things are just going to overlap and you'll form some connected cluster that fills the lattice. This is a second order phase transition generally, but again, you would not have two densities coexisting. So we really do have this first order phase transition, which is phase separation, where you have a dense gel in equilibrium with a dilute phase. People were asking how you could test this experimentally. One fun possibility is to use a relatively recent form of nanotechnology called DNA origami, where you can design these like DNA single strands that then self-assemble into various shapes that you can design from relatively simple 2D lattices to smiley faces to pretty complicated 3D objects. And so you could definitely imagine designing a magic number system because you control the interactions, you can control the shapes and the rigidity of the interacting components. And so this would actually be the perfect way to test the magic numbers. And then finally, we had a lot of really good and really thoughtful questions about choosing computational methods to study phase separation. And I would say that here, there's really no right answer, and it depends what you're interested in learning. So the magic numbers paper uses Monte Carlo simulations, which are really good for sampling the equilibrium properties of a system. So what Monte Carlo will do for you is that it guarantees that it's going to spit out states that have the right statistical properties. And so you're going to, say, measure a certain energy with the right probability. And this is great for thermodynamic properties, like getting the phase diagram or figuring out the energy of the system or what the protein cluster sizes are. But it's actually really bad for dynamics because Monte Carlo will spit out the right states, but it doesn't tell you how to get from one to the other. And in fact, you often want to take advantage of the fact that you can equilibrate your system faster by using some moves that don't make any sense dynamically, right? When you're doing polymer simulations, actually a lot of times what you want to do is force polymers to teleport from one side of the box to the other, which gives you great statistics for your thermodynamics, but is obviously not a realistic picture of the dynamics of how the system evolves in the real world. On the other hand, there's a whole field of molecular dynamics where you really write down the equations of motion, which come from Newton, and then you solve them, you just sort of integrate them forward and you see how the system evolved. So this is great for dynamics because it's just simulating the dynamics directly, but it can be trickier to extract thermodynamics, and you tend to be more computationally limited because you have to worry about how every single protein is experiencing every other protein all at the same time. On the other hand, if you have like an analytical theory of fluids that phase separate, like in a Cahn-Hilliard model, you can also just write down these differential equations and solve them numerically to study phase separation that way. And there are other, with any of these methods, I'd say that there are lots of interesting questions about the level of detail or granularity. For certain questions, you might want to do like fully atomistic molecular dynamics, where you can see what every atom in like 10 amino acids are doing for like 3 nanoseconds. But that's not going to help you if you're trying to study what 10,000 proteins are doing in a droplet. And indeed, even if you had a computer that you could do it, you might not want to because that might obscure the more conceptual questions that you're trying to answer. On the other hand, if you're trying to like build some specific design a drug or build some specific system in chemical engineering, you might really want to put in enough details that you can get a quantitatively correct answer. And so in this magic numbers paper, we were really interested in this conceptual question, which is why we made the modeling choices that we did. But I would say that in general, choosing the right tool for the job is a pretty complicated thing, and it's something that you just sort of learn through experience as you develop as a scientist. And with that, I am out of time, and we should, I guess, take any final questions. There's a question about mean field models or mean field theories. Basically, the conceit of a mean field model is that you know that like in your system there's some average, right? There's an average energy. There's an there's an average number of bonds or something, and you know that in the real world there are lots of fluctuations around that. Now, for a mean field model, you basically just sort of claim that these fluctuations average out or that they're uncorrelated, and you sort of replace you say that, oh, basically, you know, every protein has like sort of the mean behavior. Or you say, oh, instead of thinking about every protein interacting with every other protein, you can just think about a single protein interacting with like sort of an average field of all the other proteins. And so this is great because mean field models can let you learn a lot of, you know, really cool and interesting physics that can actually often get at a lot of the key questions that you're interested in, right? Flory-Huggins is a mean field model. The magic numbers theory is a mean field model. You need to go beyond mean field models when correlations become really important, and when the fluctuations away from the mean are actually really correlated, right? Like, you can imagine that, you know, if all of your proteins become unbonded, if on average, like maybe one protein gets a bond and another one drops one, and that happens at roughly the same rate, a mean field model is great. On the other hand, if something weird happens where all of your proteins fluctuate into having no bonds all at once, your mean field model is not going to capture that. It's a lot harder to do non-mean field models, and you can often learn a lot just using the mean field. Oh, there was this question earlier about the non-rigid polymers versus the U-shaped thing. And the question was basically, you know, it seems like if you have two flexible components, the entropy should be really high. So like, why are the magic numbers still relevant? And the answer is that it's not just the total entropy, it's the difference in the entropy between the dense and the dilute phase. So the idea is that, yeah, you might have a lot of entropy if you have two flexible components, but the relative entropy, the fold change in the entropy between the dense and the dilute phase is larger for the rigid components, or the dense phase is more different when you have rigid components. There's a question about the Gibbs free energy. Yeah, as we were talking about earlier, physical systems are always trying to minimize the free energy, and so phase separation happens because you can minimize the free energy by separating the two phases. That's the role. There's a question about network-based interactions. So I'm not 100% sure what this person means by network-based prediction of intracellular phase separation, but there are actually people now thinking about like protein interaction networks, right? Like, you know that like protein A interacts with proteins B and C, and there are people now who are trying to look at those network topologies and make predictions about what kind of phase separation behavior is going to happen. There's a professor at Princeton, Andre Koshmerl, who just put out a preprint about this just like a month or two ago. So that's definitely something that people are interested in. All right. I don't know. Should we call it June? Do we have anything else we need to say?
I think that's it.
All right, great.
Yeah.
Thanks, everyone. Enjoy your break. We hope you come back in an hour to hear from Ned.
Yes. Thank you very much.